Hello and welcome to another lecture of the course Aquaculture. Today we are going to discuss culture method of Mugil cephalus. Many countries have been culturing Mugil cephalus like Italy, Egypt, erstwhile Soviet Union or Russia and other related countries you can say, Israel, Taiwan, Hong Kong, Japan, Philippines, Indonesia, China, India. It is because of its wide distribution. You have studied in the previous lecture that Mugil cephalus is distributed widely across varied salinity range and across varied conditions. So um, a lot of countries, a lot of regions where the fish is found are culturing Mugil cephalus, the mullet. In 2022, global aquaculture production of Mugil cephalus was 5,972 tons. In 2022, global capture production of Mugil cephalus was reported to be much more than this. It was 1,05,818 tons. So, capture contribution of Mugil cephalus was much more than the culture of Mugil cephalus. The trend of global culture production, increased from 25,600 tons in 1997 to 1,47,000 tons in 2003. This shows that how culture was undertaken by people from different regions, and in between, say, only 5 to 6 years from 97 to 2003, there was significant increase in culture, 25,600 tons in 1997 and 1,47,000 in 2003. That was remarkable. The first country to fully complete the reproductive cycle of Mugil cephalus was Taiwan. In fact, in most of the candidate species which are not being cultured or which are not being cultivated at a very large scale or commercially, or if their culture practice is not viable, the main problem is the availability of good quality seed in desired numbers at the desired time. So, if you are having the standard technologies for induced breeding or for hatchery production of young ones of any candidate species, its culture prospects are supposed to grow faster than other species. So, that is one of the basic reasons. Global capture production in tons of Mugil cephalus. Here you can see from 1980 to 1994, there was very growth was fluctuating and very little growth. You can say that no significant growth occurred between 80 to 1994 in capture production, global capture production of Mugil cephalus. But from 94, after 94, from 1995, it was like this. You can see how the graph is going. And from 95 to, uh, you know, almost today, with few exceptions, the capture production of Mugil cephalus is quite good. And in the recent years, you can see from 2015 onwards, there has been a little decline, but it can still catch up maybe in the coming years. But we can see here that right from 1995 to say even today, with bit of fluctuations, the catch is quite good globally. And how aquaculture has performed? Aquaculture was also not that good when we were, you know, in the previous slide, we have been dealing only with the capture production. Now we are dealing with aquaculture production of Mugil cephalus in tons. And here also, until from 1980 to 1992, the aquaculture production was almost the same, but then it started to take up, and it was peaked in 1997 with very close to about 10,000 tons, and then it started to dip again, and ultimately quite fluctuating trend of aquaculture production of Mugil cephalus occurred, and currently in 2022 it is uh, just an average production of aquaculture of Mugil cephalus what we have been seeing over the years, with only exception in 2021 um, where it was close to 11,000 or so tons um, in 2021. What are culture methods? How Mugil cephalus is being cultivated? The culture of Mugil cephalus depends on choice and availability of the type of system facilities. In fact, you know, if you see my previous lectures on culture method of different species that I have talked about, and I have talked about the culture systems in general, and how they are used, what are their advantages, then you must have understood that which type of system is available at your disposal, um, you know disposal that you use for aquaculture. Maybe you can go for simple extensive aquaculture or semi-intensive aquaculture, or if you have other facilities, you may go for intensive aquaculture. There are many varieties of intensive aquaculture. So depending on the kind of facilities, the kind of resources you are having at your hand, you can decide that what kind of culture practice we should proceed with. In the traditional farming method, such as in ponds, stocking is done with fry. If well-equipped hatcheries are there, then the induced spawning method is carried out. Here the stocking starts with brood stocks. In fact, as I told you, that if you have to popularize the commercial practice of any candidate species, here for example Mugil cephalus, then the first thing and foremost thing which is required is standardizing and providing complete SOPs, standard operating protocols, for breeding, induced breeding or controlled breeding of that particular species. Or you can say that we should come up with good hatcheries for seed production or for production of the young ones of that particular species. So how we proceed in this? First, we have to um, arrange the brood stock. You can get it from natural conditions or from the pond where you have been raising Mugil cephalus. But whatever the source is, from whichever place you are getting the brood stock of Mugil cephalus, they should be this thing is taken into account. Uh, that the brood stock is mature and healthy. The health and maturity status, gonadal maturation of brood stock should never be compromised. That is one of the essential things that you have to take into consideration. So you have got the brood stock, whether from wild or they are farmed, but they should be mature and healthy. Males usually they mature at 25 to 30 centimeters standard length, not total length. You must have an idea that what is total length and what is standard length, because you will find this information in standard length as well as in total length. So please take care while interpreting the information. And age at first maturity in case of males of Mugil cephalus is usually 2 to 3 years. So you can take males of 2 to 3 years. Try to not take males of less than 2 years. Even if you notice some kind of maturity, it is not advisable to take underage males, or which in general do not mature in the age group less than 2 years. So try to take from 2 to 3 years onwards. But there are certain circumstances you can get males which may mature little earlier. But you should take into account that you have to take only those males which have passed 2 to 3 years of age. In case of females, the maturity size has been found to be 27 to 35 centimeter, standard length again. And here the maturity comes a little later in age, that is 3 to 4 years. So age at first maturity in case of female Mugil cephalus is 3 to 4 years, whereas males attain maturity 1 year earlier than the female, 2 to 3 years. In the wild, mullet maturation and spawning occur in October to January on India's east coast, and in June to July on the country's west coast. So you will have to monitor. Please do not follow generalized information, because this fish is so widely distributed throughout the world that you will get many different information on this species regarding stages of maturity and months of maturity, and many other things regarding its habitat. No, so you have to take decision based on the region where you wish to start your hatchery or culture farm. So take into consideration that, and if possible, please verify it with the stages of gonadal maturation that you can observe from the fishes of your own region. So if they show all signs of gonadal maturation, both in case of males as well as in females, then proceed in that season, in that month of the year. And generally, what we do, we try to get broodstock from wild, from natural environment, using seed nets or whatever is suitable at your place, whatever type of net you are using to catch this fish. This is, we have discussed again and again for a number of species, that transport of brooders or seed. This is very crucial in ensuring the success of your operation. So because that puts stress maybe sometimes beyond a limit that these species are unable to tolerate, and you may end up getting mortality of the species that you are carrying. Transport of fish to vehicle, plastic bags or shrimp brooder bags. You can transport it in bags that we have discussed for shrimps as well as for fishes. You can get the details in my previous lecture. Oxygenated tanks, you can use tanks directly, but the thing is that you should provide complete provision of oxygen supply. That oxygen in dissolved form is present in that tank to a sufficient quantity that is needed, or thermocol box, whatever is available. Again, I am saying you can have your own container. The thing is, the fish should not be injured in that container. There should be enough water, there should be enough of oxygen, and not so much of agitation should be there in that tank. So if these things you are taking into account, then any brooder, any fish seed, you can transport from one place to the other. Transport vehicles should contain appropriate tanks in order to occupy the bags or whatever you are carrying, container you are using for transporting these. Tank should contain water of similar salinity. That is something very important, because you should not give these fishes a kind of shock in terms of different water quality parameters. Sometimes we give them temperature shock by transferring them from water which is maybe 35 degrees centigrade in temperature, and when we are carrying them in the bag, suppose you are carrying that bag in a car which is having AC running, so maybe 25 degree temperature is there inside, and you are putting 25 degree temperature water fish into 35 degree water temperature pond, then they are bound to get temperature shock, and you may get mortality in the stock that you are carrying into your pond. So temperature shock or salinity shock or whatever type of pH shock, these things should be avoided. You must try to use the same salinity or same water from where brooders have been kept, and you are carrying the brooders from the place. Same water should be taken into your tank or bag. Dip treatment. When fish reaches site, I have also explained this in previous lecture. This applies here also, that we have to ensure that the fishes get suitable prophylactic treatment. And that prophylactic treatment may be, there are many prophylactic agents. Potassium permanganate is one of the very popular prophylactic agents which you can use. A dilute solution you can make, and dip treatment can be given to the fishes which are being carried out to your pond. Transferring the fish to the quarantine tanks for 2 weeks. This becomes very necessary, especially if you are carrying them from the wild. So you are not sure what kind of pathogens, what kind of health issues the fish may be having, and the organisms which may harm the other stock of the fish which is already there in your pond. So you must use some tank where these fishes which you have been carrying from other place to kept separately. So you have to keep these fishes which you are carrying from other place separately in the tank. Monitor them for 2 weeks. For everything you have to, this is a kind of acclimation also. Quarantine will be there, acclimation will be there to the conditions prevailing at that region. Continuous water flow should be maintained in the quarantine tank. That's what I am saying, that they will get used to this condition, as well as if there are certain pathogens or there are certain harmful conditions which can harm to the other fish stock in your pond. So all these things can be taken care of by keeping them in quarantine tanks for sufficient time. Then broodstock should be given prophylactic treatment, we have discussed, with 100 ppm formaldehyde for 45 minutes in holding tanks. When you are transferring, then you should use 100 ppm solution of formaldehyde for 45 minutes. So that kind of prophylactic treatment can be given. Then ponds, tanks, raceways can be used as a holding system. You can, depending on what kind of resources. I have been telling you in many of my lectures that it all depends on the choice of the system, the choice of the tank, that the size and shape of the pond. Everything depends on the kind of resources that you are having at your hand in your region. So you can use them as a holding system of brooders. Then continuous flow of water should be there. Stocking density less than 1 kilogram per cubic meter. That is the stocking density that we have to ensure, and the ratio of male to females will be 2 to 3 males per 1 female. And salinity 30 ppt is desirable. Feed, here in case of Mugil cephalus, we use sinking pellets so that the feed could reach to the fish rapidly to the place where it lives, and the fish can feed on the given provided food as quickly as possible in order to prevent nutrient leaching from the diet that you have given to the fish. 35 to 40% crude protein, it depends on which size, which stage you are stocking, and 4 to 8% crude lipid. Again, you can check the nutrient requirement of the species, and it's quite possible that nutrient requirement of the species is given from your region. Then it becomes more authentic. Or you can contact the agencies which are doing research on Mugil cephalus culture, or which are providing information on Mugil cephalus culture, breeding and culture, and you can come up with some very authentic scientific information which will help you out in saving your food, or increasing the efficient utilization of the food that you are offering to your fish, usually at the rate of 3 to 5% body weight twice daily. Again, I have discussed it a lot, that the amount of food, the ration level that we give to the fish, and the how many times we have to give that, split that ration in a day, the feeding frequency, that is decided according to the life stage of the fish, life stage as well as sometimes health status of the fish. Cleaning of tanks on alternate days, maintaining hygiene is of utmost important in any farming or culture practice. This is how broodstock of grey mullet Mugil cephalus looks like, and they have been kept in a holding tank. You can see here Mugil cephalus here, brooders have been shown, and you can see that I have explained it earlier also in case of calves, while I was telling you about induced breeding of calves, that one of the best indication of uh, ripe brooder that you have to select for taking induced breeding program is just take the fish, ripe fish, and here it is, just for the sake of showing you, the vent has been shown towards the camera. Otherwise, what we do, we take the fish on our uh, hand, by its belly. So by its belly, we take the fish on our in our hand, and when the belly is pressed against the hand, then through its vent the gametes may ooze out, melt, or if sometimes eggs also, a few eggs may also ooze out, or otherwise you can collect the eggs, any procedure, and observe the eggs under microscope. You can do that also. So that is one of the things that you have to observe before taking the brooder for in the breeding tank for giving injection and proceeding with the induced breeding program. So that will ensure success of your operation. Otherwise, by doing everything ultimately in the morning, by giving injection in the evening, and in the morning you are expecting some fertilized eggs there, and you don't get anything if you have not selected brooders properly. Environmental parameters that you have to take care of: temperature. The desirable average temperature is 21 degrees centigrade. It's not that you have to keep strictly 21 degrees centigrade maintained. No, that is the average desirable temperature. Photo period, less of light and more of dark period, 6 light and 18 dark it has been given. And salinity desirable range is 32 to 35 ppt. Recirculation and daily water exchange at the rate of 75 to 80%. This maintenance of water quality plays crucial role, and of course about these conditions that we have been discussing, because whenever we are keeping, taking any fish from wild and try to get it breed in the captivity, then you will have to simulate the conditions, try to simulate most of the conditions that prevailed in the wild when the fish bred in the natural environment. They do not breed in captive conditions but get mature. Maturity will be there in captive condition, but breeding usually, naturally they do not breed in captive condition. Induced to spawn in captivity, for that you will have to induce them. You can use any of the inducing agent that we have talked in detail in previous lectures. Then you can go for stripping or hormonal induction for spawning. You can allow the fish to ovulate on its own, and males then can fertilize the eggs. But the other method, the dry method, which we have discussed earlier, you can strip the male and female, and fertilization can be done with the dry method on a tray, using that bird feather dried, clean dried bird feather that I told you. That first we get milk, egg from the female, then milk from the male, and both are mixed together, and then water is added, so fertilization takes place, and then the fertilized eggs are transferred into the hatchery. Hormonal induction: purified salmon gonadotropin, human coronary gonadotropin, there are different inducing agents that we have already discussed in induced breeding lecture. So the same applies here also. You can have any of those inducing agents and try it for this fish as well for spawning, and then fertilization will occur and ultimately hatching. Reported range of temperature, salinity, time for hatching. Um, at 26 to 28 degree temperature, it has been found the salinity of 26 gram per liter, the same ppt you can say. Um, then the time taken for hatching is 30 to 32 hours when temperature is 26 to 28 degree. When temperature decreases to 20 to 24.5 degree centigrade, then and at a salinity of 24 to 0.39 to 35.29, the time of hatching increases 59 to 64 hours. And with the intermediate temperature of 23 to 24.5 degree centigrade, the hatching time is again intermediate, 34 to 38. We all know that with increase in temperature, the hatching time decreases. Sorry. With increase in temperature, the hatching time will decrease. At 20 degree, there is 59 degree centigrade, 59 hours to 64 hours hatching time when temperature ranges between 20 to 24.5 degree. But when it is increased to 26 to 28 degree, the hatching time decreases to 30 to 32 hours only. So that is common with very common thing for development that you can get in case of fishes. Stocking density of larva while going for larval rearing should be 10 to 20 larvae per liter of water. Larvae start feeding 3 to 5 days post hatching. 3 to 5 days after hatching, they will start feeding, and they feed exclusively on rotifers along with phytoplankton. So they prefer rotifers, and if you provide enough rotifers, then you can get a very good growth rate. With some phytoplankton can also be there. Algae, like what we discussed in earlier, that chlorella or nanochloropsis can be used, maintained at 5 lakh cells per ml, that can be inoculated in the culture tank, and that provides very good growth rate. The green water. Feeding 15 to 35 days after hatching, now we have to provide them Artemia apply. That gives excellent result, very good result. When we, after 15 to 35 days, we feed them with Artemia apply. Complete larval rearing time 42 to 55 days. Larvae grown up to 17 to 18 millimeter can be harvested and transferred to the next tank, the nursery tank, for their nursing. This is how fertilized eggs, larvae, and fingerlings are seen. This picture has been taken from SIBA. Production cycle of Mugil cephalus. How it proceeds. When the fish breeds, the fry collected from coastal waters and estuaries, 16 to 20 millimeter in length. Fry must go through a series of acclimatization to salinities of aquaculture system. Since this can tolerate to wide range of salinity, that doesn't mean that if you are taking say fish from 40 ppt salinity and you are transferring it to 20 ppt salinity, then that transfer should not be abrupt. There should be gradual decrease in salinity so that the fish get adapted to the salinity that you are having in your culture tank. The same thing is said here, that it should get a chance to acclimatize itself in the salinity of the aquaculture system that you are going to use. Then fry nurse to fingerlings in case of in the nursery ponds, and then fingerlings are stocked for on further growth in different aquaculture systems, whatever you are having at your hand. Grey mullet is mostly grown in semi-intensive aquaculture ponds in different parts of the country, coastal areas we are talking. And then cultured mullet is harvested at a body weight ranging from 0.75 kg to 1 kg, and they are harvested using any kind of net that is popular in different regions of the country where they are being cultured. Nursery rearing, the hatchery produced wild small fry of size 15 to 25 millimeter should not be directly stocked in grow out ponds, as the growth of this species is slow during the early life stages. To obtain the desired size of advanced fingerlings, 100 to 120 millimeters, the nursery rearing step should be carried out. So, it's always advisable to go first for nursing them separately, and when they are of desirable size, 100 to 20 millimeter, 100 millimeter to 120 millimeter, then you can transfer them in the other pond. This step can be carried out in earthen ponds, as they depend mainly on natural food. During early life stages, most of the fish species, most of the aquatic species, they prefer to feed on natural food items, the live food items. If you can provide them, if you can do monoculture of those, or if you are able to provide the production of those live food in the system itself, so whatever option you are having, you can, but try to provide them with enough live food for their proper growth. The preparation of pond is done by drying, desilting. This all we have discussed a lot. Eradication of harmful organisms and weeds, and manuring. So, preparation of pond, that we have discussed separately, as well as for while discussing culture of many aquatic species in my previous lectures. The same process you have to follow here. The only difference is that whichever region you are going to select, and what type of water body you are having, what type of topography is there, all those physical, chemical, biological factors, or abiotic and biotic factors, you can say, they decide how to manage, how to take different steps differently for that species culture. So, drying, desilting, eradication of harmful organisms, predators, removal of predators, weeds, undesirable fishes, manuring, everything you can do here also, as we have discussed earlier. Animal manure here is used at the rate of 2.5 to 5 ton per hectare. Then water is added to the pond. Turbidity of water should be maintained 25 to 30 centimeter using Sakai disc reading that we can we use for turbidity reading. Chemical and organic fertilizers are added in accordance to the to maintain the Sakai disc reading. In fact, it's not that you have to make the water turbid, you have to remove the clarity of the water of the pond. In fact, the turbidity is with reference to productivity of the pond, productivity in terms of plankton. So, if the turbidity is due to plankton, then it is always good. And what kind of reading, Sakai disc reading you should have, 20 to 30 centimeter, that is considered desirable for semi-intensive farming. Stocking density of at the rate of 125 per square meter. Supplementary feed, artificial pellets can be provided with 25% protein level. Here it is written, but that depends on the life stage, the size of the fish that you are stocking, and it's always advisable to go and check the nutrient requirement of that species. Nowadays, you have enough information from very authentic sources. You can visit the website of different institutes. You can go through the information published in reputed journals and manuals of FAO and other places. So that way you can get access to what is the protein level that is required for the food, or what are the other nutrient requirements, or maybe you can go for commercial feed which have given the proximate composition of the feed that has to be offered for which stage of the fish. So you should provide sufficient quantity of good quality, well balanced, nutritionally balanced protein diet. Culture duration 4 to 6 months according to the size of the fry. That depends on that what is the size that you have stocked and what is the size that you are going to harvest. Harvesting time size usually 10 gram is preferred. Fry size nursery rearing in Hapa 1.6 to 2.4 centimeter. Stocking density initial 30 days at the rate of 400 per cubic meter volume, and stocking density after 30 days at the rate of 200 per cubic meter, and feeding thrice a day with wet and flattened rice. Again, it's just a method you can standardize for your place, for your pond, according to the conditions, the ingredients, the diet, everything, what is at your disposal, what you can do. Depending on that, you can decide that what should be the food that you are offering, and how many times that you have discussed just now. And stocking density, in fact, if you are going to do size grading after a certain period of time, or periodical size grading is to be done, then you can have higher stocking density initially. Otherwise, go for the stocking density which is suitable for culturing the same fish in the same pond. So, depending on the conditions, you can have your own decision. Grow out culture, how we do in case of ponds. Pond preparation, first pond preparation should be done, that we have discussed at length. And animal manure is used 2.5 to 5 tons per hectare. Water level depth about 1 to 2 weeks for phytoplankton development, 25 to 30 centimeter. So, first we are preparing the pond, keeping the water for proper phytoplankton development, for the development of production of the live food that should be utilized by the fishes. Water level then raised for fingerlings to 1.5 to 1.75 meters. Then fertilization through manure and chemical fertilizers to maintain 20 to 30 centimeter Sakai disc grading, in order to ensure that the productivity of the pond is not compromised. Fish stocking size 10 to 15 centimeter size is preferred, that is desirable. Stocking density at the rate of 6175 to 7410 per hectare. But you know, again, I should say that depending on every factor that you are having at your hand, there is always scope of flexibility, little bit flexibility at your place. Feeding rice and wheat bran are preferred, but here rice as well as wheat bran, none of them are considered as good protein source. They are in fact energy rich ingredients. So, you must include some protein ingredients also. For example, if you have to provide traditional food, go for maybe rice bran or wheat bran, whatever is available at your place, or rice polish, whatever is there. And then you can have oil cakes, any of the oil cakes, groundnut oil cake, then soybean oil cake or mustard oil cake. So, that will be a good protein source. After proper processing of these ingredients, prepare the feed, and according to the feeding habit, you can offer the feed to your cultured species. Culture period 7 to 8 months, and then after 7 to 8 months, we are supposed to get 4.3 to 5.6 tons per hectare per crop, 4 to 5.5 you can say tons per hectare per crop that we expect with this kind of culture. Fingerling of Mugil cephalus, how they look like. Then, very interesting thing is that we can do semi-intensive culture, polyculture with this mullet, and see the combination mullet, carp, tilapia. So, they are compatible, they are compatible species, although from very diverse habitats they have been taken into one pond for polyculture. What is the stocking density? Stocking density of mullet is given here, about 2.5 you can say thousand, or 2470 to be more specific. What is the reported value? 3705 per hectare. And carps 1850 to 2470 per hectare, 100 gram size. Tilapia 61,750, it's basically a tilapia pond, to 74,100 per hectare, 10 to 15 gram size. So, uh, three type of fishes you have, you know, stocked in polyculture, and total yield is 20 to 30 tons per hectare per crop. Mullet yield is 2 to 3 tons. So, this is, I should say, a very good development in case of polyculture of mullets along with carp and tilapia. So, all these three species, they are very popular as food in humans, and they have high market value. So, you can get yield of very good quantity of all these species. Cage culture, cage culture concept we have already discussed. So, cage installed in the brackish water system, stocking size 10 to 15 grams of mullet you can stock, and density at the rate of 27 numbers per cubic meter. So, if we are using this 10 to 15 gram size, then 27 number of these 10 to 15 gram size per cubic meter should be the stocking density. For culture duration, we go for 195 days, and grow out size 380 to 550 grams has been reported. Um, you can go for any kind of culture system that we discussed in the beginning only, that depending on what kind of system you are having at your disposal, just by your own wisdom, your own intellect, you can decide that which one to select, which one to opt for. And depending upon the conditions, you can go for even biofloc culture here in case of Mugil also, Mugil cephalus. Alternative, efficient and sustainable system, we have discussed a lot about biofloc. So, again, I am not going to explain it further. This is quite efficient that we have discussed. You know, a lot of advantages the biofloc system is having. Continuous nutrients recycling and reuse, that is one of the best thing about biofloc, that here you take help of microbes, and they do every kind of cleaning and nutrient recycling in the system itself based on the growth of microorganisms. Minimum or zero water exchange, that is again the beauty of this biofloc system. And microorganisms uptake nitrogen compounds and convert them into microbial protein that we discussed earlier, and thus maintain water quality. And this protein may also serve as food to the organisms that you are culturing in biofloc. So, and this is supposed to lower the input feed cost, and provide better performance of the fishes which are being cultured in biofloc. Microorganisms help in culture feasibility, reduce FCR, food conversion ratio, and decrease feed cost. So, the points that I was just talking about. High density fry rearing can be done in this system, that we all know. This is one of the modern culture systems where we get very good production, and this is considered as eco-friendly as well. So, biofloc culture can also be undertaken. So, that's all in this culture of Mugil cephalus. Thank you so much.